Hello, welcome to this session all about SCAR20, that is SCAR20. It is my absolute pleasure to introduce both of our speakers today. First off, so we have a double feature for you today. First, we have Dr. Mike Henne from UT Southwestern. Dr. Henne received his BS in Cellular and Molecular Biology from Texas Tech University in Lubbock, Texas, and then accepted an MRC Scholarship from the United Kingdom to pursue a graduate studies at the MRC Laboratory of Molecular Biology at Cambridge University. Following graduate school, Dr. Henne began a postdoctoral position at the laboratory of Scott Emr, PhD at Cornell University, as a Sam and Nancy Fleming Research Fellow. Dr. Henne's lab at UT Southwestern exists at the interface between lipid metabolism and human diseases. His lab has pioneered studies of cerebellar ataxia disease, including, in particular, SCAR20, and we're among the first to report the cellular basis for this neurological disease caused by a loss of function mutation in the gene SNX14. And our second speaker today is Dr. Naiara Akizu from the Children's Hospital of Philadelphia. Dr. Akizu received her PhD from the University of Barcelona for the discovery of the epigenetic chromatin modifications that regulate nervous system development. During her postdoctoral studies at the University of California, San Diego, in the laboratory of Joseph Gleason, Dr. Akizu applied genome sequencing of large cohort of patients with neurodevelopmental disorders and uncovered the genetic causes of a handful of cerebellar ataxias, including Spinocerebellar Ataxia Recessive type 20. And with that, I'll turn the floor over to our speakers. The floor is yours. Welcome.
Very good. Thank you so much, Celeste, for this introduction and everyone for joining the webinar. Today, Mike and me are going to tell you how our independent interest in SCAR20 merged several years ago to establish a fruitful collaboration that is quickly advancing our understanding of a new group of Spinocerebellar ataxias and a protein family with exciting novel functions that are associated with the pathogenesis of SCAR20. And I will start this presentation by telling you a little bit about how and when we discovered the disease and what are the genetic causes. But before I tell you this, let me just briefly introduce what are the characteristics of SCAR20, what is the pathology and clinical features. SCAR20 or Spinocerebellar Ataxia Recessive 20 is a neurodevelopmental degenerative disorder that is usually diagnosed during the first 2 years of life due to a suspected global developmental delay, including delayed motor learning or speech learning that are detected in young children. However, what it makes SCAR20 different to other neurodevelopmental disorders is the presence of smaller cerebellum early on, like in these two children diagnosed with SCAR20. This smaller cerebellum is usually caused by a perinatal onset and progressive degeneration of cerebellar tissue that leads to the smaller cerebellum with the extra space between the cerebellum and the skull, like you can see in these two patients, in contrast to a full cerebellum that fills the full posterior part of the brain in a healthy child magnetic resonance image. In addition to the smaller cerebellum, patients with SCAR20 develop intellectual disability and a characteristic facial feature or facial morphology that is characterized by a deep nasal bridge, upturned nose, enlarged forehead, and enlarged lips also. And this combined smaller cerebellum, facial features, and intellectual disability is already are already clinical signs that lead the clinicians to suspect of the diagnosis of SCAR20. In addition to these characteristic features, patients with SCAR20 or half of the patients with SCAR20 also show autistic-like features, and then features like cardiac defects, urine oligosaccharide and sugar abnormalities, larger liver and spleen, or skeletal deformities that are usually seen in other type of disorders that we know as lysosomal storage disorders. These disorders, in general, are usually a group of disorders that you will understand a little bit better later when Mike explains the mechanisms, how they link to SCAR20. But several years ago, to try to identify the cause of SCAR20, we recruited 111 families with kids, at least one, that showed a smaller cerebellum in the magnetic brain imaging and signs of cerebellar ataxia. In addition, these families had some sign of recessively inherited disorder. So just to recall, recessive disorders are disorders that are caused by carrying two copies of the mutated gene, like in this affected child. And these two copies of the mutated gene comes usually one from the father and one from the mother, who are generally healthy because father and mother, in addition to the mutated gene, they also have a copy of the normal or healthy gene that allows them to function normally. And this group of disorders that we know as recessive disorders are ideal for a successful identification of genetic causes when we do genome or exome sequencing. Here I am just summarizing what are the major findings when we do exome sequencing or genome sequencing in an individual proband. In this case, we did exome sequencing in these 111 families that had kids with cerebellar ataxia. And usually when we do this, each individual shows initially like 20,000 variants compared with the reference human genome. In order to know which of these 20,000 variants are the cause of the disease, we apply several filters, including quality filters of the sequencing analysis. We also look if the variants are present in healthy population. If they are present, then we filter them out. And then we use also protein damaging prediction programs to identify those genetic variants that are more likely to affect protein function. And this usually narrows down to 100 variants per proband. That's still a lot. But in recessive disorders, where two copies are needed to be mutated to cause the disease, when we apply the recessive disease filter or this like biallelic filter, we can narrow down to about 5 variants per individual. And then if we analyze parents that need to be heterozygous or additional siblings that have the disease or don't have the disease, and how these 5 variants are segregated in the families, we are typically able to narrow down to 1 to 3 variants per family or individual, which leads us to a successful identification of the genetic cause of a recessive disorder. And as a summary, when we did this exome sequencing in the 120 or 111 family and their members, we were able to identify novel genetic causes in about 1/4 of these families and the affected kids. And among these novel genetic causes, SNX14 here was one of the most recurrently affected gene or more recurrently mutated gene. We identified exactly 22 patients that were coming from 12 independent families, all with mutations in SNX14 and smaller cerebellum, intellectual disability, and characteristic facial features. We published this at the end of 2014, and in parallel, Philip Steiner's group in England also published other 3 families with mutations in SNX14 and very similar clinical features supporting SNX14 as the cause of SCAR20. Since then, there have been additional 40 patients reported in the literature, and there are many more in the world. All these 40 patients that have been reported in the literature carry 2 mutated copies of SNX14, as it is a recessive disorder. And these mutated copies usually lead to a premature stop in the protein, which results in loss of the protein function. As you can see here in this type of assay that we usually run in the lab, where patient cells, here in P, don't show any signal of SNX14 as compared to the unaffected family members that have some signal of SNX14, indicating that the SCAR20 is actually caused by loss of SNX14 expression. And in parallel to this identification of the gene, Mike was working on SNX14 and cellular function, and I'm going to turn the control now to him to tell you more about the mechanism and function of what is SNX14.
Thank you, Naiara. That was a great introduction. And I want to thank Celeste and NAF as well as everyone here for inviting us to tell you a little bit about our research and a little bit about how far we have come understanding SCAR20, the mutant gene SNX14, which Naiara beautifully introduced, and what we're doing right now to try to further understand this disease and ultimately develop therapies and treatments for it. So like Naiara said, while she was doing her pioneering work linking these mutations in SNX14 to this cerebellar ataxia, our group was also working on this disease, although we really didn't know at the beginning that we were working on this disease. And you'll see in a few minutes how our lab's interest, I'm a professor at UT Southwestern in Dallas, how our interests in this disease and this gene, sorting Nexin 14 or SNX14, kind of met up with Naiara's interest in studying this disease. And we now have a very fruitful collaboration and are really working ultimately to understand this disease to develop treatment. So what I want to do to tell you a bit about how this disease actually occurs on the molecular length scale is answer 3 questions. So one is, where does this protein that Naiara introduced, SNX14, that is mutated in this disease, where does it function inside of our cells in our bodies? And related to that is what cellular processes does it function in? And that, of course, will give us insights when this protein is mutated into what goes wrong in our cells, of course, that lead to SCAR20. And I'll touch on our current working models at the end of my section on why we think mutation in SNX14 leads to SCAR20 disease. But to begin, you saw from, as Naiara kind of lined up how she conducts her science, she works with patients. We also work with patients in my lab, and patients sell some, but we're a very basic cell biology lab. So we work with a variety of model systems to try to understand human disease. Believe it or not, one of the most useful model systems we actually use in my group, and one that actually gave us some really important insights, I think, into SCAR20 is the model system on the screen, which is budding yeast. This is an organism. It's also known as Saccharomyces cerevisiae. You may actually know it as baker's yeast. And this is actually the exact same organism. If you're going to go in your kitchen later and bake bread, it is baker's yeast, Saccharomyces cerevisiae, that you are going to use to do that. This is also the organism that allows us to ferment wine and ferment beer. So it has many uses, and it turns out in science labs like ours, we actually use it to study human genetics. And that is because just like our cells, budding yeast, baker's yeast, are unicellular eukaryotes. They have many of the genes that human cells have, and they have many of the cellular compartments, organelles, that human cells have. And so we could actually use them as a very easy to culture, very easy to genetically dissect and understand model system to ultimately study human genetics like Naiara's group and the basis of human disease. And it's really our journey in our group to study SCAR20 that began in budding yeast about 10 years ago, right after I started my research lab at UT Southwestern. I had a postdoctoral fellow in my group. You could see a picture of her at the bottom of the screen, Hannah Hariri. Hannah has since graduated from my lab and is now a professor with her own lab in the US. But she was studying a protein in yeast called MDM1. And what you're looking at on the left side of the screen is the gene structure for the protein MDM1. Genes often encode proteins, of course. And if you look carefully, you could tell that these different boxes in MDM1, these are the different domains encoded in the gene. It's very similar to the SNX14, sorting nexin 14 gene that Naiara introduced a few minutes ago. And that's because these genes are homologs. They're essentially have a common ancestor, and they are very similar to one another because they have similar functions either in yeast cells or human cells. And so we were studying MDM1. No one really knew what this gene or the protein encoded by this gene did. But Hannah, when she was in my lab, made a really interesting observation. When she went and imaged where the protein MDM1 actually localized to inside of yeast cells, and that's what you're looking at in the middle of the screen. This is a fluorescence microscope image of the inside of a yeast cell that we've labeled different compartments in the yeast cell with different fluorescent labels. It turned out that MDM1, which we've labeled here in green, you could actually see the protein here in green, it localized on the surface of a really important cellular compartment, and that is the lysosome, which is here in blue. Lysosomes are stomach-like compartments, stomach-like organelles for our cells. Our cells have lysosomes, and they are extremely important, of course, to do basic functions for our cells. They break down cellular debris, and they break down many of the lipids in our cells. Folks in the audience are probably familiar with different lysosome storage diseases, and it was really exciting when we learned that MDM1 actually localizes not just to the surface of the lysosome. Hannah actually found that it localizes to a particular spot on the surface of the lysosome, and that is where the lysosome contacts another really important cellular organelle, and that is the organelle in red here, called the endoplasmic reticulum, or the ER. And this, again, is another cellular organelle that our cells have. All of your cells in your body have an ER network, an endoplasmic reticulum, and this is a biosynthetic organelle. This is where many of the lipids that your cells need to live are actually manufactured. And Hannah, and she published a few papers in my lab studying this, what she found was that yeast MDM1, this gene that looked so similar to sorting nexin 14, it was basically a bridge protein. It was what we call an interorganelle tether, a bridging factor that connected these two cellular compartments. And she actually worked out on the molecular level how it tethered these two cellular compartments together. You could see that at the very beginning of the protein for MDM1, there are these two black lines. These are transmembrane domains. They are anchored in the membrane of this red endoplasmic reticulum. And then this protein reaches out just like a bridge and actually touches the surface of the lysosome with this blue PX, we call it a Fox homology domain, that binds to specific lipids that are decorated on the surface of the lysosome. This was exciting to us because there weren't that many proteins known to connect organelles like this together, to bridge these organelles together. And we had actually discovered a new one in yeast that was a relative of the sorting nexin 14 protein. Now, while we were doing this, as Naiara mentioned, she was independently doing her pioneering work with SCAR20 patients and ultimately discovering and publishing in her paper at the end of 2014 that mutations in sorting nexin 14, this gene that really no one knew what it did in the human body, led to SCAR20 and was characterized, among other things, and we'll talk about this more in a few minutes, the death of specific neurons in the cerebellum, Purkinje neurons, which are these large, very important neurons for cerebellar function. And a key thing that Naiara discovered in that paper, not only that this mutation in this gene was causative of SCAR20, but was that one of the problems in the cells when mutations were occurring in SNX14 was the lysosome was having problems functioning. And you may remember our work in yeast found that MDM1 was associated with the lysosomal compartment. And in fact, we made similar mutations to what Naiara was doing with her human experiments and found that also our lysosomes and our yeast cells were also malfunctioning. And so that was, I think, a key connection that our two groups made early on, was that this protein family, the yeast homolog, MDM1, and then human sorting nexin 14, had some relationship to lysosome biology. And so a graduate student in my lab, Sanchari Datta, when she joined my lab, she was really interested in this question of, well, does human sorting nexin 14, this direct homologue of our yeast MDM1 protein, does it also function as a bridging protein, as an interorganelle tethering factor? And long story short, she published a few papers in my group here, basically finding that very similar to what we found in budding yeast, human SNX14 in human cells also acted as an interorganelle tether, as a bridging factor that had a very similar mechanism to connect the endoplasmic reticulum to lysosomes and other organelles as well. We actually find under certain metabolic conditions, sorting nexin 14 can connect to other compartments like lipid storage compartments called lipid droplets. But it suggested that the function of this protein was conserved between yeast and even human beings. And I'll just take a second and say that other labs throughout the world have worked on closely related proteins to SNX14 and also found that they are these molecular tethers, these bridging factors that can connect organelles together. And I want to highlight beautiful work done from Suzanne Pfeffer. She's a professor at Stanford who published a paper a few years ago showing that SNX13, which you can see here, has a very similar architecture. It's a very similar protein to SNX14. And we'll hear in a little bit from Naiara again, who will tell us about, we think, some disease links to SNX13. But Suzanne's group a few years ago published a paper that found SNX13 also is a molecular tether that has a very similar function connecting this ER network, this lipid synthesis compartment, to lysosomes and endosomes, which are very similar to lysosomes in our cells. And lastly, I'll mention work from Juan Bonificino's group, a researcher at the NIH, and his postdoc, Amra, who's now a professor actually in Toronto at the Sick Kids Research Institute. She's a good friend of mine. She did pioneering work on SNX19, again, another relative of SNX14, showing it also acts as a molecular tether. So an emerging theme is these proteins are connecting proteins. They connect the lysosome to other organelles like the endoplasmic reticulum. And so we've answered our first questions. Where does SNX14 function in cells? It seems to localize at interorganelle contacts and has a particular interaction with these digestive compartments in our cells known as lysosomes. So let's then move on and ask, what cellular processes does SNX14 function in that could give us insights in how mutation in it leads to SCAR20? And so I mentioned a few moments ago, my graduate student, Sanchari, she's since graduated from my lab and is now a research scientist at the Broad Institute at MIT. But while she was in my lab, she really wanted to learn more about how mutation of SNX14 led to cellular pathology. And I'm boiling down a lot of the observations that she made, both in our lab and also collaborating with Naiara's group and Phil Stanier's group in London. But to boil it all down, what she found was SNX14 is very important to control excess lipids and what we call lipotoxicity in our cells. We all know that we ingest lipids, or you may know them as fats, as part of our diet. Our bodies also manufacture lipids. They're a key component of our cells. But if we have excess lipids in our bodies, particularly certain types of toxic lipids, this can lead to cellular lipotoxicity and ultimately to death of cells, particularly neurons. And what Sanchari's experiments, what her studies really suggested was SNX14 not just acted as a bridging factor, connecting lysosomes to other compartments, but it also played an important role in suppressing toxic accumulation of excess lipids. And one of the ways we know that are some of the assays she was actually able to do with patient cells that were actually very generously donated from SCAR20 patients throughout the world. So you're looking at two assays that Sanchari did in our lab. These are called lipotoxicity assays, where she's culturing human cells in experimental conditions, either wild-type cells that have a functional copy of SNX14 or mutant cells that do not have a functional copy of SNX14. And what she found is if she exposed these cells to different lipids, here she's adding a toxic fatty acid lipid called palmitate, that they died. They were much more likely to die from exposure to this toxic lipid compared to wild type cells. You can see that both in our knockout cells that lack SNX14 or these SCAR20 patient cells right here. And that really suggested that accumulation of toxic lipids inside of cells that lack SNX14 led to cell death. while we were doing these experiments in parallel, Naiara's lab was developing a mouse model to be able to further understand the molecular basis of SCAR20 disease. And she's since published some work on this mouse model. We helped her a little bit, but her lab was definitely the driver of this work. You could see on the left here, this is a mouse that Naiara's group was able to genetically delete the SNX14 gene. And it turns out that very similar to a human SCAR20 patient, it manifests essentially all the major symptoms of SCAR20. One of those major symptoms, like I mentioned a few minutes ago, is the progressive death of these Purkinje neurons in the cerebellum of the animal. You could see these Purkinje neurons as this layer of red cells here in these control cerebellar samples on the top of the screen. And in our SNX14 knockout mouse, you could see those Purkinje neurons are gradually dying. A key thing that Naiara's group was able to do is ask what is happening to lipids. We were finding in cultured cells that the SNX14 knockout cells were sensitive to toxic lipids. They also had imbalances of specific lipids in those cells. Naiara's group was able to use lipidomics of cerebellum samples to find that they were also manifesting defects in lipid metabolism. And together with some of our data, really suggested that SNX14 was really important to maintain lipid homeostasis inside of cells. And so a model began to emerge. Not only was SNX14 a tethering protein, could connect organelles together, it was really crucial for cells, and we'll tell you more in a moment, particularly neurons, to maintain lipid balance. And loss of SNX14 really was leading to cellular lipotoxicity through mechanisms that at this point we didn't completely understand, but knew it was tied into lipids somehow. So now in the last few minutes, I want to tell you why we think losing SNX14 adds to lipotoxicity. And to do that, we're gonna talk about the atomic structure of SNX14. So at the atomic level, understanding what a protein looks like in our cells is probably one of the best ways for us to get molecular insights into what goes wrong when there are mutations in this protein. And work that our group has been doing together with Naiara, but also a structural biologist, Brett Collins, you could see here at the bottom, has given us in the last few years a really nice atomic insight into what SNX14 looks like and how it interacts with lipid metabolism. So this was work that Brett at the University of Queensland in Australia was leading together with a student in his lab at the time, Blessy, who then, after she graduated from Brett's lab, moved to my lab at UT Southwestern. And what Brett and Blessy were trying to do was elucidate the molecular architecture of SNX14. We've been showing SNX14, Naiara and I, as this linear cartoon of this kind of long beads on a string-like protein, but it turns out in actual reality, inside of our cells, proteins take on very complex molecular structures. They're very large macromolecules composed of thousands of atoms. And researchers like us have been able to use artificial intelligence in the last few years to really elucidate what proteins look like at the molecular level. One algorithm, an AI algorithm called AlphaFold, has really been game-changing for our community to understand what proteins look like and therefore what mutations in those proteins do to their function. And so using AlphaFold and also some biochemical approaches, Brett and Blessy, together with our group, were able to get a pretty accurate depiction of what SNX14 actually looks like. So you're looking at the AlphaFold predicted structure of SNX14 on the screen. You could see I've color-coded these different box-like domains, the same color they are in the cartoon. And what I want you to appreciate is there's a surprising interaction that we only would have discovered through this AI technology, and that is that this orange domain that we call the PXC domain is folding back and touching this green PXA domain. And again, we would not have expected that just a priori from looking at an amino acid sequence of this protein, but the AlphaFold really suggested that these two protein domains form an extensive interaction with one another. And when we look more closely into the structure that they form, and this is a video that I'm going to try to play. Toggle the structure back and forth and show you that this green domain and this orange domain heavily interact with one another, and they form essentially a tube. And I'll show a still version of that tube right here. So this is a picture of that green and orange domain where we've now resolved all the atoms that are basically composing that domain. And what I want to point your attention to is this cavity, this tunnel, this tube-like structure running through the entire length of the domain. These orange residues that are lining the tube are very hydrophobic, and that means that they love to interact with lipids. And if you may recall, we found that SNCS14 has some role in lipid metabolism. What we've done here is actually model a phospholipid, a specific type of lipid, in this cavity. And what we found from this, what we think we are finding, is that this is a lipid-binding cavity whose job is to bind lipids and to transport lipids across different compartments in the cell. You could see a cartoon version of what our model is from this right here. What AlphaFold essentially told us is not only is SNCS14 linked to lipid metabolism, it's probably a lipid transport protein, a new type of lipid transport protein whose job is to move lipids from one compartment to another compartment. And that may very well be why it is a bridging protein connecting these two compartments together. And so AlphaFold is an artificial intelligence software package, of course. We need to use experiments in the laboratory to actually test whether this prediction is true, whether this protein SNCS14 actually encodes a lipid binding module. And a student in my lab, Caitlin, has been undertaking this. This is part of her PhD dissertation. And I'm happy to say that so far, her experimental data is confirming what we see using this AlphaFold AI algorithm. So what you're looking at on the screen is a lipid binding assay. We have purified that region of SNCS14, which you could see here. And the goal was to mix that purified protein with fluorescent lipids, just like what we're seeing in this cartoon, to see if it binds those lipids and ultimately can move those lipids from one region, one membrane compartment to another compartment. And she's in the middle of doing these experiments. But this assay, this native gel binding assay, essentially tells us that yes, this protein does seem to be able to bind to lipids, specifically phospholipids. And that was really exciting to us. I remember when Caitlin brought this data to me because we found earlier that it was really mutations in SNCS14 that were specifically altering phospholipids, the specific type of lipid in SCAR20 patient cells. And it really suggested that that may be because of losing the ability to bind and transport these lipids. And so you may be asking yourself now, how can this lipid binding, this lipid transport activity really help cells in our bodies, really help neurons in our bodies, and how could mutation of SNCS14 lead to the SCAR20 lipid metabolism defect? And so in the last few minutes, what I want to tell you is our working model and new exciting data that Naiara's group and our group have been compiling in the last several months that really suggests that this lipid transport function of SNCS14 is important for lysosomes to maintain their lipid integrity, their membrane integrity to function in cells like neurons. So if you imagine lysosomes, I've introduced them as, of course, digestive compartments. It's where cellular debris is delivered to be broken down in our cells. They're essentially bags of digestive enzymes inside of our cells, and they have a membrane bag-like outer layer. And it turns out that over time, the wear and tear of functioning as a digestive compartment will break down that membrane, will cause what we call lysosome membrane damage, which will make these lysosomes leaky. They will begin to leak out these digestive enzymes, and ultimately that could lead to cell pathology and cell death. This is particularly true in very long-lived cells like neurons, which you have your entire adult life. So you need to maintain the lysosome membrane so that lysosomes can function and help the cell function. In the last few years, there's been emerging basic research that has suggested that one way that lysosomes membranes are maintained is to repair these micro holes, these micro tears that are formed over time. And it seems that lipid transport proteins like we think SNCS14 is, are important for delivering fresh lipids to the lysosome membrane to essentially heal this damage. And so we have been testing this hypothesis that SNCS14 is essentially aiding in lysosome function by repairing lysosome membrane damage. And one way that we could do this in a controlled laboratory setting is we could actually induce lysosome membrane damage ourselves. We do that in the laboratory by adding chemicals that damage lysosomal membranes. One of those is called LLOME. And what we find, you could see some representative pictures of it here on the left, is when we add LLOME, we induce lysosome membrane damage. And in fact, when we do this in cells that don't have SNCS14 or this other very close relative gene, SNCS13, the lysosomes do not repair themselves as effectively. There's actually a defect in lysosome membrane repair when we have mutant or depleted SNCS14 or SNCS13. And this is new data that's coming in from folks in my group, Caitlin, who I mentioned, and Emma, as well as Vanessa, who's a really talented graduate student in Naiara's lab at her laboratory in Philadelphia. To go even a step further, we're doing this in human cells, but Vanessa and Naiara's lab is actually doing this in modeled neurons. And what you're looking at on the screen now, really exciting new data, I think, are cultured neurons that have been differentiated. You could see they have very neuronal-like morphologies, and they are expressing fluorescently tagged SNCS14 or SNCS13, and they have been this LLOME drug has been added to them to induce this lysosome membrane damage. And what Vanessa has found, very excitingly, is SNCS14 and its close homologue SNCS13 seem to sense this lysosome membrane damage and actually become recruited to these lysosome compartments, we think, to help begin to heal the lysosome membrane damage that the LLOME is causing. You could see that recruitment with these donut-like enrichments of green SNCS14 or green SNCS13 that are accumulating following adding that damaging agent, LLOME. So altogether, this really suggests that a role for SNCS14 is in lipid metabolism. In particular, we think it's in lipid transport, but how neurons really benefit from that is it may be able to move lipids to compartments that are undergoing membrane damage, like lysosomes. And in SCAR20, our working model that we're continuing to test is that when you mutate SNCS14, you lose this damage control factor. Lysosomes are still going to be damaged, but the healing repair pathways basically are not working properly. And so over time, neurons, particularly those Purkinje neurons, are going to accumulate damaged lysosomes, eventually cause those lysosomes to not work. And we think over time, that is why those Purkinje neurons may die, leading to many of the symptoms of SCAR20 disease. And so that's where we're at. This is after about 8 or 9 or more years of active basic research. I just want to say that we've come a long way. And like Naiara, I think, mentioned a little while ago, one of our big drivers to do this is to understand ultimately how this disease emerges so that we could develop therapies. And I think Naiara is going to talk a little bit about some recent findings that shed some light on potential therapeutic intervention here at the end. But I'm going to hand the talk over to her right now so she could talk a bit more about diagnosis.
Very good. So just in a couple of slides, I told you initially or at the beginning how the disease is clinically diagnosed based on clinical features, right? But our genetic findings led to a more precise diagnosis by genetic screening of mutations in SNX14. And actually, there are now several, I have problems, but here we go. There are now several ataxia genetic panels that are used to screen for mutations in ataxia-causing genes when there is a family history of ataxia or when there are signs of cerebellar or spinocerebellar ataxia. You can see here one of them is from Flugent, and SNX14 gene is included within this panel. However, what happens when all of these genes are negative after running these panels? So one option in these cases is to run exome or genome sequencing like we did with our initial 111 family cohort. And when exome or genome sequencing is run, then new genes or new mutations can be identified that are not yet included in these genetic panels. And by doing so, we have actually been able to identify a new cause of a SCAR20-like disease. And surprisingly, or maybe not, this new cause is mutations in SNX13. Just a few years ago, we were contacted by Karen and Alexandra in France, in Paris, telling us that they had 2 siblings with a smaller cerebellum, cerebellar ataxia, intellectual disability, and dysmorphic features very similar to those we find in SNX14 mutant patients. And these 2 siblings had a loss of function mutation in SNX13, inherited from healthy parents that were heterozygous for the mutation. And just in the last few years, we have been able to identify additional patients coming from Joe Gleason's lab and Fosun's clinical testing group, also with very similar cerebellar ataxia and SNX13 loss of function mutations. So this is a good example on how our functional work that we run not only in SNX14 but also in MDM1 and SNX13 and other family members can actually confirm or help establish a new group of SCAR20-like diseases and a new genetic diagnosis too. And in the lab, we are now doing some few experiments to confirm that actually mutations in SNX13 can lead to the disease. And Vanessa in the lab has generated SNX13 loss of function human pluripotent stem cells, differentiated them to neurons, and then analyzed lysosomal features like lysosomal lipid content, in this case LOD4 stands for lysosomal cholesterol, and as you can see here in green or here in black, the amount of lysosomal cholesterol in SNX13 knockout neurons is lower than in the control neurons. And similar to what Mike has shown in cellular cultures here, when we treat SNX13 knockout neurons with LLOME, these neurons have also problems repairing lysosomal membranes, suggesting that actually SNX13 and 14 loss of function lead to lysosomal membrane repair problems that likely underlays the pathogenesis of SCAR20 or SCAR20-like disorders. So what about the treatments? Unfortunately, we don't have treatments or curative treatments for SNX14 and SNX13 type of diseases currently. All that is out there is just management tools to support patients with specific clinical features. However, we think that our work can help us understand a little bit better how we can intervene to prevent the neurodegeneration. And actually, not just our work, but recent work that was published using another SNX14 knockout mouse model suggests that treatment with valproic acid might suppress cerebellar degeneration in this mouse model at least. So this is the knockout mouse that shows loss of Purkinje cells, here labeled in brown. And this is the knockout mouse treated with valproic acid, showing less degeneration of Purkinje cells. And although we still don't know how valproic acid suppresses cerebellar degeneration in SCAR20 mouse model, we know that valproic acid is used as an anti-seizure medicine, and it's not very well understood either how valproic acid inhibits seizures. There are some studies suggesting that this inhibition is related with the inhibition of breakdown of GABAergic inhibitory neurotransmitter. However, what we think is that valproic acid might also change lipid metabolism in neurons, helping Purkinje cells or neurons survive better, because we know that valproic acid also affects lipid metabolism and lipid storage in lipid droplets. So Mike and I are now planning to study how valproic acid might be affecting all these like lipid metabolism and lysosome ER and lipid droplet interactions, and trying to identify additional mechanisms for or additional strategies for the treatment of patients with SCAR20, hopefully in the future. And with this, we would like to finish concluding that SNX14 and its homologs are molecular interorganelle CTs. SNX14 low sensitizes cells to lipotoxicity. SNX14 contains a putative lipid transport domain. SNX14 is recruited to damage lysosomes and might play a role in membrane damage repair. And with this work that we've been doing in the last 10 years, very exciting and collaborative work, we hope to keep understanding better the mechanisms of SCAR20 and hopefully lead to alternative therapies that can improve the condition also.
Thank you so much, Dr. Akizu, Dr. Henne. So I already see one question in the Q&A window. For some context, we recently had a webinar on terminology around ataxia diagnosis information. So things around ARCAs versus SCARs. So I will refer people to Dr. Leanna Rosenthal's talk on this subject, but how these are both describing recessive forms of ataxias, but they're slightly different systems for organizing them. But we have an attendee who asks, does SCAR20 have a corresponding ARCA number? And then if you could give your thoughts on the ARCA versus SCAR numbering system, if you have any you'd like to share.
I don't think SCAR20 has a corresponding ARCA number, no.
Yeah, I haven't seen that either.
Yeah.
I believe that the gene was discovered after the ARCA system has been started to be phased out. So some of the, shall we say, older or more, the less newly discovered or named forms of autosomal recessive ataxias, they'll have usually both an ARCA and a SCAR. But since SCAR20 was more recently described, then it doesn't have a corresponding ARCA number. But thank you for asking. And just while we're waiting for some questions to come in, a question from me. Could you share a little bit more for us about, you talked a little bit about lipid metabolism, lipids in the cells, for folks who aren't as familiar with what a lipid is and what role it plays in the cells that keep us all alive and going. Could you talk to us a little bit more about what these are and what they do in the human body?
Yeah, that's a wonderful question. Maybe I'll field that one. So lipids are a basic building block of our cells. So we talked a lot about lysosomes today. That lysosome compartment itself is really composed of lipids. Lipids we can think of as fats. We of course ingest them in our diet, but they're really just a hydrophobic molecule that makes up the membrane compartments. It really is a lot of our cell. It creates the boundaries of our cells. But lipids can also be used for other things too. They're used as an energy source. They can be burned to harvest energy, and that's why we, of course, many of us store too many of them. If we've had maybe a few too many hamburgers over the years, but they are a fundamental component of our bodies and neurons in particular. Long-lived cells, being able to maintain their lipid metabolism is critically important because these are cells that really you have from birth to death. But lipids. We talk about lipotoxicity. Lipids can be damaged over time, and they could also be chemically modified and become very toxic for our cells. So being able to maintain lipid balance, we call it, or degrade toxic lipids, we now are, I think, collectively knowing is a major, a very important basic aspect of human health and leads to a lot of diseases, not just neurological diseases like scar diseases, but metabolic diseases as well.
Thanks for explaining that. Do you have anything you'd like to add?
Yeah, one more thing. Something surprising that I found when I started working with lipids is that the brain is actually the second most lipid-rich organ in our body after the fat. So
yes,
yeah, that shows how important lipids are also for the brain, right?
Definitely, that underscores just how important it is for, well, all the thoughts and thinking that people are doing on a day-to-day basis.
Mm-hmm.
Well, thank you so much for explaining that and clarifying. For folks who maybe they have a loved one who, after hearing this talk, they think, oh, maybe it's SCAR20, or they already have a family member who is diagnosed and they're concerned for other family members and they'd like to learn more. Do you have any recommendations of places they can go for more information on SCAR20 or resources for people who want to get a little bit of assistance with that diagnosis?
Yeah, so unfortunately, I don't think there are that many resources to learn more about SCAR20. There is this human gene website that is collecting the genes that have been identified in neurodevelopmental disorders, and I know that SCAR20 and SNX14 is one of the genes, and they are basically collecting genes and patients and clinical features all the patients. But other than this, I don't think there is any other resource. And also, unfortunately, there is not much known about SNX14 and 13, right? So you go to Google or PubMed and type SCAR20 or SNX14, and there are just maybe 20 papers or less that come up. So yeah, I think that's why this type of webinars are really useful, and I'm happy to answer or meet with anyone that wants to know more. I'm sure Mike too. I do this for other disorders also, and I think my email is somewhere there. Definitely.
Yeah, I want to reiterate that as well. I mean, if you have any questions, if you want to get connected, reach out to Naiara or to I. I also talk a lot to the Niemann-Pick community. Niemann-Pick type C is, of course, another type of neurological disease. And they have a similar foundation to NAF called the Ara Parsegian Foundation. And really, a lot of patients first get to know about that community by contacting a researcher. They may hear about something from an online webinar or hear about research. So reach out to one of us, and we'd be happy to connect you to any communities that we know, because often researchers like us do know not just communities related to a specific disease, but we know other related disease communities as well, and we can help connect you.
That's so helpful to know. And if any of you are watching this recording in the future or currently present and are having some issues finding Dr. Akizu's or Dr. Henne's email and would like to be put in contact with them, please feel free to email research@ataxia.org. We will pass along your message. And perhaps time for one last question. You mentioned how we don't know a whole lot about SCAR20 at the moment, but more research is being done to progress that knowledge and find out more information. If there are people on this call who would like to get involved with that research process to help move things forward, what kind of options are available? Is it best to contact researchers like you?
I would say, yeah, that's probably the best.
That's a great start, right? Contact researchers like us. Hopefully, we can answer any questions you have, I think, first and foremost, and kind of tell you where we're at as a community, but also connect you to, I think what NAF is doing and other similar organizations, the Parsegian Foundation I mentioned. They are really, trying in many ways to equip patients and patient families with information, with avenues sometimes to donate support these research efforts, but it often starts with just an email or a phone call. So feel free to reach out.
Well, thank you both again for joining us today and sharing some of that information with the members of the NAF community who have SCAR20 and people who care for them as well. So on behalf of the National Ataxia Foundation, thank you both so much for presenting with us. We really appreciate your time and your expertise.
Thank you to you. Has been a pleasure.
Same. Yeah, thank you so much.
And for everyone who's on the call who spent the last hour with us learning more about SCAR20, I hope you learned something new about this condition and something useful. And wherever you are in the world, good morning, good afternoon, good evening. Have a wonderful rest of your day. Take care, folks.